Hey guys, welcome back to One Stop Biology. So guys, in today's video, we are going to continue with the chapter molecular basis of inheritance. In the last video, we covered translation, a very important process in terms of genetics, right? So I hope you would have undergone the previous videos that I have made for this chapter. And in today's video, we are going to basically discuss the regulation of gene expression and how important it is with respect to all the different other processes that we have studied. So, guys, before we start off with the video, I would definitely request you to give your feedback at the end and definitely request you to like the video, share it with your friends, and do subscribe to the channel if you are new to this channel so that, I get to know that yes, you are liking the videos, or do let me know over comments of this video that you didn't like anything in particular, or do you want, if you want to improvise on something. So because of the help, because of the feedback that I'll be getting from your videos, basically I'll be improvising, and it'll help me a lot, to structure the videos properly as per your convenience. So guys, let's start with regulation of gene expression. Now basically it refers to a very broad term, right? Now, because regulation of gene expression can occur at various levels, right? Because that gene expression results in the formation of a polypeptide, right? Ultimately. So, again, if you remember the central dogma, you have then DNA, from that you have RNA, and from that you have proteins. And then this DNA keeps on replicating. So, you have replication, you have transcription, and you have translation, right? So with this, for going through different processes, you have proteins formed. So the gene expresses itself in the formation of proteins or polypeptides. Now it can be regulated at several levels, right? So in the case of eukaryotes, this regulation can be exerted at first transcriptional level, right? So when the primary transcript is getting formed, you can regulate the gene. Then processing level. So when you are splicing that transcript, you can basically regulate the expression. Then when you transport that RNA from nucleus to cytoplasm for translation to happen, right? So when you transport this mRNA from nucleus to cytoplasm, again, gene can be regulated. And then at the very end, during translational level, when translation is happening, one can regulate the gene expression, right? So basically, what we can say, guys, in terms of functioning of a gene is that the gene in a cell, the genes are expressed to perform a particular function or a set of functions, right? For example, say there is an enzyme which is known as beta galactosidase. There is an enzyme which is known as beta galactosidase, right? Now this is synthesized by E. coli. Right. So this beta galactosidase is used to catalyze hydrolysis of disaccharide, lactose into galactose and glucose. Right. Now the bacteria E. coli use them as a source of energy. These lactose and disaccharides, when broken down, they are used as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source. Basically, they would no longer require synthesis of this enzyme, right? Therefore, we can say that it is the metabolic, physiological, or environmental condition that regulate the expression of a gene. So basically, it is expressed when it is required metabolically, physiologically, or as per the environment conditions, right? Now, in the case of prokaryotes, what happens in the case of prokaryotes? Control of the rate of transcriptional initiation is the prominent site. Remember, guys, which control of the rate of transcriptional initiation is the predominant site for control of gene expression. So basically, in a transcription unit, the activity of RNA polymerase at a given promoter is basically regulated by interaction with accessory proteins, right? Now, this affects its ability to recognize start sites. And these regulatory proteins can act both positively, as in they can act as activators, and they can act negatively, as in they can act as repressors, right? Now, the accessibility of promoter region of prokaryotic DNA is regulated by, in many cases, basically, it is regulated by interaction of proteins with sequences termed as operators, right? So this app operator region is adjacent to the promoter element in most operons. And in most cases, the sequence of the operator binds a repressor protein. And each proton has a specific operator and specific repressor, right? So we'll take an example and we'll study this operon basically, which is Lac Operon, right? So Lac Operon is present only in, basically Lac operator is present only in Lac Operon, and it interacts specifically with Lac repressor only, right? So let's see what exactly Lac Operon is. So guys, basically Lac Operon, was found by an association. So there was a very close association between some geneticists, so which were Francois Jacob and a biochemist, which was Jack Monod. So I'll write it again. Who? Francois Jacob, who was a geneticist, and there was a biochemist, Jack Monod, right? So they in close association basically elucidated the Lac operon. Now they were the first to elucidate a transcriptionally regulated system, right? Now in Lac operon, where Lac refers to lactose, remember, Lac refers to lactose. What happens is that a polycistronic structural gene. So I hope you would have, if you would have, seen the last few videos, you would understand what a monocistronic and polycistronic gene is. So basically, in Lac operon, a polycistronic structural gene is regulated by common promoter and regulatory gene, right? Now, such arrangements are very common in bacteria and it is known as operon, right? So you have Lac operon, you have TRP, tryptophan you operon, you have ARA operon, you have his operon, you have Val operon, right? So a lot of these kind of operators basically are present, whether promoter or repressor, and you have so many regulatory genes basically which regulate the polycistronic structural gene, right? So the Lac operon consists of one regulatory gene which is known as the I gene, which is basically the inhibitor gene, right? I gene, which is the inhibitor gene, and then three structural genes X, Y, and A. Now, I gene basically codes for repressor of Lac operon. Again, I'll repeat. I gene codes for repressor of Lac operon. Z gene codes for beta galactosidase, codes for beta galactosidase, right? Now, this is primarily responsible for hydrolysis of disaccharide lactose into monomeric units like galactose, glucose, and all that, right? Then you have Y gene. Y gene codes for permease. Now, this permease increases permeability of cell to beta galactosides. Then we have A gene. A gene encodes for transacetylase. A gene codes for transacetylase, right? Now, all the three gene products in Lac Operon are required for metabolism of lactose. Remember that, guys. All three gene products in Lac Operon are required for metabolism of lactose, right? So, if you see here, if you see the diagram as well, this is the basic Lac Operon. This is the basic Lac Operon. Okay, now let's see the functioning. So what happens is that basically lactose is the substrate for the enzyme beta galactosidase, and that regulates switching on and off of the operon. And that is why it is termed as inducer, right? So that is termed as inducer. Now, in the absence of a preferred carbon source, such as say glucose, if lactose is provided in the growth medium of the bacteria, lactose is transported into the cell through the action of permease, right? Now, this lactose that then induces the operon, and how it induces the operon? So, the repressor of the operon is synthesized. First, from the I gene, repressor is synthesized. So, you have basically this is the I gene here, right? You have repressor mRNA, and from this repressor is synthesized, right? Now the repressor protein binds to the operator region of the operon. So this basically arrow, as you can see, this binds to the operator region of the operon, and this prevents RNA polymerase from transcribing the operon. This basically restricts transcription. Now, in inactivation, right? So basically, in presence of this inducer, such as lactose or allolactose, the repressor is inactivated. Remember, the repressor is inactivated by interaction with the inducer. Now, again, that allows RNA polymerase access to the promoter and transcription proceeds. So, essentially, regulation of Lac operon can also be visualized as regulation of enzyme synthesis by its substrate, right? So, once the inducer comes, so basically, this is the inducer. So, what I said that in presence of the inducer, the repressor becomes inactive. This is an inactive repressor. And as it becomes inactive, you have transcription, right? So transcription is happening, which is then translating. So translation is happening, and again you have beta galactosidase, you have permease, and you have transacetylase, right? So basically, this is in absence of inducer like lactose. This is in presence of inducer like lactose, right? And here what is happening in absence, this repressor is getting transcribed, and this repressor binds to operator region and prevents RNA polymerase from transcribing the operon, right? So basically, regulation of Lac operon by repressor is referred to as negative regulation. It is known as negative regulation. So Lac operon, the regulation of Lac operon by repressor is known as negative regulation. But Lac Operon basically is under control of positive regulation as well, right? So basically, right now, for you guys, you have to just understand working of the Lac Operon, right? And I hope you understood it completely with the help of this video. Guys, if you didn't, please do let me know of your doubts either in the comment section or please message your doubts over WhatsApp and I'll clear it out as soon as possible. But yes, if you did understood the video very well, please do not forget to like it, share it with your friends, and yes, if you are new to the channel, again, I would request you to subscribe to the channel for all the updates in future. Thanks for your time, guys. Bye bye.